Good morning, everyone. My name is Devin Fishburne, Marketing Manager at MDIC, and I'm here to wish you a very warm welcome to this edition of MDIC Live, a fireside chat style series highlighting the latest resources, projects, and happenings within the Medical Device Innovation Consortium. Our next guest was recently named Director of MDIC's Clinical Diagnostics Program, and she's had a very busy week, I must say. First, she presented on MDIC's early success with its Somatic Reference Samples project at the Next Gen Clinical Diagnostics Conference. Then she worked on the launch of MDIC's new initiative within SRS, and now she's here to tell us more. This guest has covered a lot of ground to spread the word about the importance of this project, so let's give a warm welcome to Maryellen DeMars.
Hey, Devin, it's great to be here and have the opportunity to talk about the Somatic Reference Sample Initiative as well as the Clinical Diagnostics Program at MDIC. So glad to be here.
Good morning, Maryellen. Thank you for joining us. And for those of you that are joining us via Facebook, Twitter, YouTube, and LinkedIn, of course, we want to remind you that this session is interactive. So if you have questions for Maryellen, please feel free to post them in the chat. Let's keep the conversation going and feel free to collaborate with our colleagues in the comment section. Without further ado, Maryellen, let's get started.
Okay.
So the first question is an easy one. Tell us about yourself and your experiences professionally, both prior to MDIC and now.
Happy to do that. So I'm going to tell a story that dates myself, but the fact is we're all getting a lot of experience. So when I was in high school, when I just started out in high school, I was in biology class and we learned about DNA. And in my head, a light bulb went off and I said, that's what I want to do. I didn't know what that was at the time, but in fact, I am doing that and working very closely with DNA and looking at mutations and reference samples to help support the sequencing efforts. So I had the opportunity since high school to work for a number of organizations, both for-profit and not-for-profit, and in a variety of roles. My background was science. I went for a PhD in molecular virology, but then went into different companies in industry as well as not-for-profit. And most recently, my passion and my experience and my focus has really coalesced around reference samples and diagnostics. I've had the opportunity to work in a organization, not-for-profit, that provided reference samples for diagnostic partners. I worked in a diagnostic company. We were developing a COVID diagnostic needing reference samples. And then I also worked for a startup that was focused on external and independent evaluation of diagnostics to support reimbursement, regulatory, and adoption. So having the perspective, of all those, kind of aspects of diagnostics, I feel very fortunate that I am kind of living my dream here. That I have my passion is the reference samples for diagnostics, and the opportunity came up to lead this SRS effort. And since then, I have taken on the expanded role of program director for clinical diagnostics, and really happy to have ability to have a broader impact in this area.
Right. So it sounds like the dream continues. I mean, you're taking on an expanded role at MDIC as the program director for clinical diagnostics. So you're taking SRS project and kind of expanding it even further. Talk to me about your goals in that role. What are you looking forward to?
Yeah, so what we're really focused on is projects. SRS is one of them, and we'll be talking more about the details of that, but also other projects that will help improve diagnostic testing and product development using novel regulatory approaches through collaboration. I think the aspect of MDIC that has worked so well and is a great model is that we're the convener of collaborators, and we're able to really pull strong teams together to drive results. And our ultimate goal with the clinical diagnostics program is to provide predictable pathways for innovative diagnostics to get to market. And there's always going to be new technologies, new challenges, new diseases that need to be addressed kind of on the fly in kind of the medical arena. And so we are providing a way to address these new challenges and new diagnostics, new technologies in a way that makes the getting them to market in more flexible efforts and adapting to what is being developed in the medical community.
Yeah, that's great. And for, to level set for a second, for anyone that's not familiar with the topic, let's take a step back and talk about actually what are somatic reference samples. Can you give us an overview for someone who's, new to the subject?
Yeah, this is a great question because I must say somatic reference samples probably 100 times a day because this is my world. But let's take that term and divide it into somatic versus reference samples. And let's start with reference samples. Reference samples are kind of a generic term used to describe something, an entity that's characterized either qualitatively, quantitatively, or just it's a known entity. So it's been evaluated and it's known, and that is used to validate tests. And so we're not only talking diagnostics, but reference samples are used in clinical chemistry and different areas. It's a known quantity. So when you run it through your machine, you know what the answer should be. And if the answer is not correct, then you have to address an issue. So that's what really reference samples are. And then the somatic reference samples that we're talking about, I'm going to go back to some terminology, germline versus a somatic. Germline mutations are ones that are inherited in our DNA. So they're there from birth, and they are the genes that we live with. Somatic mutations are ones that have been introduced post-conception. And in this case, we'll talk a little bit more about it, but in the case of cancer, there are a lot of genetic mutations that occur that actually are the basis of the disease. So what we're trying to do is to construct these reference samples that have known cancer mutations in them. And then we can use these constructed somatic reference samples as a method for validating and hopefully and eventually supporting regulatory approval using these samples as tools.
And these samples, to my understanding, have the potential to be tools in a lot of different areas of medicine and of healthcare. But one of the ways we've seen them be kind of used more commonly is in research around cancer and cancer variants. So with the knowledge that these genomic tests are important, or these somatic references are important to finding cancer variants, especially in clinical trials, can you outline, further, how do these somatic reference samples come into play and how are they used in,
Sure.
the field?
Sure. And I'm going to bring this down to kind of unfortunately, an everyday occurrence is, if you or a loved one or a friend, somebody in your world is diagnosed with cancer, often they'll go and they'll get a biopsy. And then that biopsy will be taken and then evaluated and tested. And one of the tests that is done now and more frequently is next-gen sequencing, to look at the genetic sequence of that tumor to see what changes have occurred. And if those changes, there's a potential that those changes that have occurred actually have a therapy that can be used to address that disease based on that genetic variant or those genetic variants. So these reference samples come into play because, as you can imagine, the accuracy of the test to determine the sequence, the accuracy has to be incredibly high, because if there's even a single nucleotide that is misrepresented or misreported, then you might not be getting the right therapy, or you might not be having your disease addressed in the right manner. So the accuracy of these tests is incredibly important. And with these reference samples that are a truth sequence, that it is known now what that, if you use those reference samples to test on your diagnostic platform, you know the sequence of the reference sample, you know what should be reported after the test is done. And if there's an inconsistency, then there's an issue with the accuracy of that test. Now, the way that this comes into play in clinical trials is that's right now, that's an avenue, and the only avenue, the best avenue for some of the cancers that are detected. So there is chemotherapies that are associated with cancers. There are standard of care, but really there's an emerging area of drug development, and the only way to access these is through clinical trials. And you're included or excluded for clinical trial potential therapy based on your genetics. And so you have to make sure that the genomic sequence that's reported through the next-gen sequencing-based diagnostics is correct in order to get into the right clinical trials and be exposed to possible beneficial therapy.
That's great. And you talked about all the ways that these somatic reference samples can be used a little bit in your presentation at the Next Generation Diagnostics Conference, where you discussed the pilot project from MDIC to develop, manufacture, and create these validated data sets for an initial 10 reference samples. Can you give us an overview of that project and tell us a little bit about what you covered in your presentation?
Certainly. So this project was started to be envisioned in 2018. And it was interesting that I was actually, I was in a different position and working in an industry organization. And I was in the original working group for the, at that time was called Cancer Genomics reference samples. And I was able to see and help, kind of form the project that came to be. Then I stepped away and did other things for a while that took me out of participating in that group, and then able to come back and lead this. So what was defined in this project, it originally started that we had identified 54 cancer variants that we wanted to construct into reference samples. And then through more discussion and looking at funding opportunities, we were able to narrow it to a pilot project, which I actually think is a great approach. Let's do the pilot project and then take those lessons learned and expand it. So we narrowed it down to 10 cancer variants. And we have a manufacturing partner, Horizon Discovery, that is actually doing the construction of these reference samples using CRISPR technology. They're taking a highly characterized human cell line that has been the subject of a lot of work at NIST. And they're then engineering in 10 cancer variants, one into each cell line. So at the end, we'll have 10, the same cell line that 10 different clones or whatever that have the different variants in them. And then we will use those as standards and reference material for testing the accuracy of NGS platforms. And the other aspect of this, it's not only the samples, but also the data sets. We're working with NIST as our partner doing the bioinformatics, and they're generating these validated data sets. And together, the data sets and the samples themselves are tools that can be used not only by diagnostic developers, but also have the potential to become tools to assist in the evaluation of the regulatory agencies and approval by regulatory agencies. So the power of these samples is quite incredible. And this is a way, since next-gen sequencing is such a sophisticated technology, how to fully evaluate these for regulatory approval has been a challenge. And these samples are helping address that.
And you mentioned that these samples have the potential to have so many different uses. They can be so powerful in their application, but they come with a lot of challenges. And you mentioned some of them being, low availability, what are some of the challenges that the lack of established, accurate community samples presents in work like this?
Yeah, so one of the challenges of reference samples has been the replenishment potential and the availability. So in the past, one of the sources of reference material has been clinical samples or discarded clinical samples. You know what the diagnosis is, you know what the sequence is, and you can use those as the reference sample as you're doing new clinical testing. The issue with that is that that's a finite resource. And so it's been a struggle, as long as I've been in reference samples and standards, it's been a struggle to find a mechanism or a pathway to generate consistent batches of replenishable material. So that's what we're looking at as we're introducing these mutations into cell lines. That cell lines are replenishable. You can store them and then you can grow them as needed. And theoretically, they're a resource for the foreseeable future. One of the issues is you have to make sure that there's going to be consistency in the batch to batch, and that's one of the other things that we're looking at is looking at multiple batches of these reference samples that are being generated and making sure that we understand any changes, anything that could impact the consistency and the utility of these reference samples. So again, I'm going back to the fact that this is a very NGS, next-gen sequencing, is a very powerful technology, and again, very sophisticated. So needing to look at ways to assure the accuracy of these. And so this is with this approach of replenishable, and the fact that we're going to be doing studies on the consistency of batch-to-batch variation, I think has the opportunity to provide a very powerful tool and a more consistent and available tool than looking at or waiting for discarded clinical samples.
And how can MDIC's work within this project look to address some of those concerns around this reproducibility, the sensitivity, and the accuracy of these samples?
Yeah, and I think that comes from the collaboration aspect of this. The fact that we are working with a very strong manufacturing partner who their world and their focus and strength is reference material. So they're very familiar with this world and they understand the importance and have the background to support this project. The fact that we're working with NIST as the key bioinformatics lead on this project, the fact that we're working with the FDA on our steering committee to help guide, this as a potential tool to help regulatory approval. The fact that we're all working together and we have a clear vision on what these samples are and what they can do and how they can be used, I think is remarkable. And this all comes from the amount of characterization that is involved in this project, both from the manufacturing, the QC, the looking in detail at what has been created and making sure those changes are in the right, that when we're creating these mutations, that they're in the right place. And there aren't any other changes that we're introducing, making sure that they're, that the cell lines are growing and replenishable, and then looking at, the amount of characterization of the data sets, the intensive, a bioinformatics analysis that's going on makes the pairing of the data sets with the samples a very strong tool.
And it sounds like you guys have, a lot of work on your hands. It sounds like you guys have a lot of things to kind of collaborate on as well. And you have a lot of help in doing that. You mentioned working with FDA, and you mentioned at the top of the show, working with Horizon Discovery. And MDIC recently announced that partnership with Horizon Discovery on this SRS initiative. Could you tell us, how did that partnership come about? And what are your goals in forming collaborative partnerships like this one?
Yeah, and again, they're a critical partner in all of this. So when this effort was being developed in the 2019 2020 timeframe, it became very evident that we would need a partner for the manufacturing. And so part of the approach was to send out an RFP to look at saying this is what's needed, this is the type of partner that we want to work with, and get potential organizations to respond to that. And so we did get a few responses, and Horizon Discovery was selected based on the responses to their RFP. And and this year we were able to actually sign the agreement and get this going. And cell lines are growing, and we started the actual technical work. But I can't emphasize enough how much Horizon, me being in the industry and in this area of reference samples, they are a known leader in this, and they're involved in a lot of different efforts, and it's an important part of their business. So I was really pleased when I came on and that this partnership had already been established. They're a fantastic partner. And so what we're looking at is right now we're doing that we have the 10 variants, but then there's a potential with them to do even more. They have the technical capability to do that. And one of the challenges for us is going to be to get additional funding to support that. But we have a potential long-term partner here that can stay with us throughout the effort, whatever that size and shape it comes to be.
Right. And I mean, speaking of whatever it comes to be, where does SRS go from here? And how can those of us that are watching the broadcast on LinkedIn, Facebook, Twitter, and YouTube get involved and learn more about this project?
Yeah, so that's a great question, and it's something I think about almost every day, because as we're doing this, I continue to see more potential. And one of the goals right now is the 10, right? And the 10 reference samples that we're doing as the pilot project. And then through those efforts and lessons learned, we're still very interested in that expanded project where the initial goal was to do 54. And so I think there's a number of ways we can go. One is let's get keep going with the early successes and get the lessons learned. And then we can expand into the additional 44 variants that we've identified. There actually might even be more now since we did that landscape analysis and that survey. So we'd have to relook at the variants that are associated with cancer. But also it's applicable in other areas as well. And so if you can think about that, any diagnostic test that is using next-gen sequencing as a diagnostic, this could apply to, and that includes hereditary diseases or infectious diseases. So there's a potential to expand into that area as well. And then the other direction to go in is that the driver for these reference samples were certainly from the regulatory aspect. Can we make tools that will help validation and regulatory approval? And that's really the original driver. But you can see that these tools have application and utility in the entire product life cycle. One is the early on doing the feasibility studies or the actual development of the product. But even after product launch, in addition to the regulatory, in product launch, you can look at these, or could these reference samples be tools to support reimbursement decisions? So that's an area that I want to get into as well in terms of, let's start talking. We've had a few conversations, but is there, again, supporting regulatory, is there an opportunity to support, post-launch and post-market efforts as well?
That is wonderful, Maryellen. And hello, everyone. My name is Deborah, and I'm going to be stepping in for the Q&A section of today's session, as well as providing an announcement for today. So, Maryellen, thank you so much for the overview of the SRS initiative. I am very grateful that you had the chance of walking us through all of this. Now, the main question that I have for you is how people can interact with SRS in the future. What engagement opportunities do they have available?
Yeah, and that's a great question too. So right now we the activity that is focused on through a steering committee that is all our partners in this, the FDA, different government agencies, diagnostic manufacturers, and funding agencies, and so that's really kind of driving the project right now. But what I see in the future is first, if people are interested or in this field and interested in this from the sample perspective, or you're in the diagnostics manufacturing business, or whatever aspect of this, that you can go to the website and you can look into not only SRS but also other of the clinical diagnostics programs that we have going on, and so then also if there's any programs or projects that people feel that have synergy with what we're doing, then I would encourage them to get in touch with me at MDIC, because it's really valuable to have looking at other projects, looking at other efforts, and getting input and insight and lessons learned from similar projects. So we're starting to those discussions and partnerships with external organizations that might be doing something similar, so that we can all learn together.
Great, and that is wonderful. That's what we want to do. We want people engaging with your project at different stages. So thank you so much for that response. Now we have received some questions in the comments, and I would like for us to kind of go into a Q&A. And if somebody else has any questions with the 3 minutes that we have left, please send them our way. So the first question that we have is data from NGS sooner getting regulatory approval for medical device claims?
So I'm trying to understand the question. So is the data from NGS getting regulatory approval for medical device claims? So in some cases, the answer is they're going for that. And we're trying to, it's going to be a while. This project probably isn't going to have the samples out until 2024, but we are trying to get these as tools to make that easier in order to get those claims for regulatory approval for NGS.
Wonderful. Thank you, Maryellen. And then we have another question of how can we help fund your project?
I like that kind of question. So one of the things that I didn't really speak about, but what we're trying to do is really have an awareness campaign about this. You heard Devin at the beginning talk about the presentations we had, when we're trying to get the word out there as much as possible. We have been successful in getting philanthropic organizations and diagnostic developers and manufacturers to support this pilot project. And then, but we're currently looking at, ways to increase the funding by this awareness campaign, and trying to show the impact of what we can do and how this could help the entire medical device industry. So I think the idea is that, we're starting some internal efforts to outreach for funding, but certainly if anybody is interested in this project and from a financial support perspective, would love to talk to you.
Wonderful. And the other question that I have connected to that, is there any opportunities where people can connect with you? Do you want to share your contact information or the page where they can find more information about your initiative?
Yeah, certainly. I can follow up with that. Absolutely. Yep.
Wonderful.
Happy to do that.
Do you have any closing statements that you would like to share with your audience today?
Well, the only thing that I can say is that I continue to be extremely excited about this, and happy that I came to MDIC at the right time, that this opportunity was here to continue the work that I had started when I had worked on the working project. And then now the expanded opportunity to really affect the development and hopefully expedited entry to the market for some of these innovative technologies. So look forward to working with the medical device industry, with our collaborators at MDIC. And the other thing is that we have some other very interesting projects that are on the horizon that are evaluated by a steering committee. And I'll be able to talk about those more once they get initiated. But there's a lot of exciting things on the horizon that we're going to need participation for, and organizations to join us and support some of these exciting opportunities.
And is one of those upcoming exciting opportunities participating in the annual public forum for MDIC?
Yes, it is. So that's a good point, Deb, that we are one of the concurrent sessions that we are presenting at the annual public forum, which is September 13th for MDIC, is that we will be talking about the kind of what's next, what's in the future, what the goals for the clinical diagnostics program at MDIC. And I have some great panelists that are joining us, FDA, NIST, device manufacturers, and looking for an exciting conversation.
I'm looking for that conversation as well. So Maryellen, thank you so much for joining us this morning and sharing more about your incredible initiative. I am very happy to have learned more about that, and I can't wait to catch your session at the upcoming annual public forum. So thank you so much for joining us today.
It was my pleasure. Glad to have this opportunity.
And for every one of you who have connected with us every week for the MDIC Lives that we have had them on, thank you for your continued support, and we hope to see you in the next sessions of MDIC Live. See you later.